Hey everybody! Today we're going to be doing a transformation lab. Well, you're going to be doing a virtual one, and I find that in labs students really struggle to understand what's going on the microscopic level, so I usually do a puppet show for them. And so for this lab, the video you're going to make is a puppet show yourself. I want to see that you understand what's actually going on in this lab. So here's my puppet show. Alright, so we start off with some E. coli cells. These are not very special E. coli cells. These guys, they're pretty vanilla. They don't do much, alright? But we want to make them cooler. We want to insert some recombinant DNA. Some DNA that we have made that has some cool new genes on it. And we're going to give these guys a cool new ability, and that ability is going to be that they get to glow in the dark green. Alright, so we'll set those aside for a minute, and I'm going to show you this plasmid. This is the plasmid that we're going to be using. And I meant to draw this out on paper, but I'll just show you on this piece of paper here. Alright, so this plasmid has several parts. Can I line this up? Yeah, I can. Okay. So the green part represents the GFP, the green fluorescent protein. This is a jellyfish protein that allows jellyfish to glow green. And this green fluorescent protein happens to be controlled by this purple gene here, ARA-C. And ARA-C is a gene, it's a gene that's induced by arabinose. And it turns on the GFP gene. So in other words, we have built a plasmid where we can turn the GFP production on and off simply by adding arabinose, which is a sugar. So we can control whether the bacteria are actually producing GFP or not. And then this gene right here, I call it AMP-R because it's for AMP resistance, but it's actually called the BLA gene, BLA gene. And it encodes for a penicillinase that chews up ampicillin, which is a beta-lactam drug. And so bacteria that get this plasmid become resistant to ampicillin. This is technically an R factor that we have designed, custom designed, to contain a resistance gene, a GFP, a green fluorescent protein gene, and then this regulatory gene that turns the whole thing on, turns GFP production on when we add when we add arabinose, sugar. The ampicillin gene is always on. So bacteria that have this plasmid will always be ampicillin resistant. Okay? So in lecture, we watched a video of some cells that are naturally competent and that have their own fishing equipment to go fishing for DNA in their environment. Well, these E. coli are not naturally competent, but we can make them competent using a little trick in the lab. And that is, we take a cold ice bath, all right, and we add some calcium chloride. This is just sodium chloride, but calcium chloride, which is a salt, to it, all right? And then we soak the E. coli in this cold, salty bath, and we add the plasmid, all right? So the salt is supposed to supposedly neutralize the DNA and make it easier to take up by the cell. DNA is negatively charged. So supposedly it neutralizes the cell, neutralizes the plasmid, makes it easier to take up into the cell. So another thing we have to do in order to encourage the cells to take up this DNA is something called a heat shock. So what we're going to do is we're going to take these out of the ice bath. We're going to get a hot water bath, well, warm water. We're going to dip them in here for about 50 seconds, about a minute. And then we're going to take them out and put them back into the ice water bath. We call that a heat shock. So you go from cold to warm to cold. And when that happens, they take up the plasmid. Not all of the cells will, but some of the cells will take up that plasmid and become transformed. And so the cells that become transformed now have this plasmid. They've taken up the plasmid, and we say that they are P-glow positive. The plasmid is called P-glow. All right. Some of the cells, though, they don't end up taking up that plasmid, and we call them P-glow negative cells. Lost my piece of tape. So we've got a combination of P-glow positive and P-glow negative cells. So now these are in our tube. We can't see them. They're microscopic. We want to grow them, right? So we're going to put them in culture. Just going to like prop them up back here. All right, we're going to put them in some media. There's my little P glow minus. Okay, we're going to give them some media. And a classic general media that we use is called LB, or Luria broth. So this chicken soup, all right, chicken stock is going to represent my Luria broth. So given Luria broth, both of these types of cells will grow. They're going to look the same on the media. We're going to have no idea which is which because we haven't given them any arabinose. They're not going to be producing the green fluorescent protein. So they'll both grow, which is annoying because then we're just going to have a bunch of cells and we're not going to know which ones have our plasmid and which ones don't. So the next thing we're going to do, on the next plate, we're going to give them some ampicillin. So the LB broth, to the LB broth, we're going to add some ampicillin. Here's my ampicillin pills for us, okay? Now, the only cells that can grow in the presence of this antibiotic ampicillin are our P glow positive cells that have that plasmid with the ampicillin resistance on it. So these guys will all die. And on media that has LB and ampicillin, the only thing that's going to grow is our transformed bacteria, our P glow positive E. coli. All right. Now, if we want our P glow positive E. coli to actually make GFP, we need to add arabinose. And arabinose is a sugar, so I'm pretending to add a spoonful of sugar. A spoonful of sugar helps the medicine go down. Oh, my little singing. Okay. And so when that happens, now, wait for it, here comes the magic. All right, now our cells will glow green. Ta-da! End of puppet show.